My name is Tom Parlapiano, and it's an honor to welcome you on behalf of our MLK Event Planning Committee. We're so grateful that you are choosing to gather with us today. We want to celebrate and appreciate the many organizational partners and sponsors who have made this year's celebration possible. Thank you to our founding partner, the Connecticut Department of Energy and Environmental Protection. And thank you to the New Haven Museum for co-hosting the celebration this year. The Peabody's 26th annual celebration of Dr. Martin Luther King Jr.'s legacy of social and environmental justice is generously sponsored by citizens, with additional support provided by the Office of the Secretary and Vice President for University Life and Belonging at Yale. Just a few housekeeping notes. Closed captioning is available for this program, and this feature can be accessed through the meeting controls at the bottom of your screen. For any questions during the program, please use the Q&A feature, which we will monitor throughout the program. You can also use the chat feature if you have any observations to share or are having technical difficulties. We want to begin the program by acknowledging the current and ancestral stewards of the land we are calling in from. Yale University acknowledges that Indigenous peoples and nations, including Mohegan, Mashantucket Pequot, Eastern Pequot, Shagtacoke, Golden Hill Pawgasset, Niantic, and the Quinnipiac, and other Algonquin-speaking peoples, have stewarded through generations the lands and waterways of what is now the state of Connecticut. We honor and respect the enduring relationship that exists between these people and nations and this land. For today's program, I'm pleased to introduce Yara Alshwairikh. Yara is a PhD candidate at Yale University School of Environmental Studies. She is interested in ecology and evolutionary biology and has a special interest in reptiles and amphibians. For her dissertation, she is exploring the phylogeography of wood frogs and their remarkable adaptation to extreme freeze tolerance. Yara will talk about her work on genetic variation in this species and its incredible tolerance for the cold, which we're going to be experiencing later this weekend. With a few specimens from the Peabody's collection that have been essential in her research, Yara will walk through how museum samples are used to generate molecular data in the lab. With that, I'd like to introduce Yara.
Thank you, Tom. Hi, everyone. Good morning. My name is Yara Alshwairikh. I'm a third-year PhD candidate. Thank you all for coming to the session today. I'm very excited to talk to you about frogs. So when I do any kind of talk, I always like to start with a little bit about me, just to tell you how I got to where I am today and how I got interested in this field. So I have here a map of the world, and I've highlighted in red stars a few relevant locations. So I grew up in Saudi Arabia, on the Middle East, and I grew up in a large city, and I didn't have many opportunities to experience nature and wildlife. I then moved to Los Angeles, California, where I did my undergraduate degree and I studied biology. I then went back home to Saudi Arabia, but this time to the West Coast, where I did a master's degree program and I studied marine science. And finally, in 2019, I moved back to the United States, this time to the East Coast, and I started my PhD program in 2019. Next slide, please. So I know this is a talk about frogs, but I have to talk about lizards just a little bit because this was my first experience working with reptiles and amphibians and doing any kind of wildlife research. So it wasn't until my third year in college where I had this opportunity to really experience wildlife and just the whole experience of doing a research project. I had this chance to do a field biology course with a small class. And the theme of that class just happened to be reptiles and amphibians. This wasn't something that I had an interest or passion for before that experience. So my group mates and I designed this project. We were especially interested in the western fence lizard and the abundance of ticks on their bodies. And I really like to mention this experience because I think sometimes when we talk to researchers and scientists, especially in wildlife, we have this image that they've always been immersed in that field. And like when they were kids, they were running around chasing bugs and other animals. And I just wanted to highlight that that is incredible. And a lot of these stories are really great. But there are different pathways to science and like finding this passion. And I only developed this interest towards the end of my college years. Next slide, please. All right, so now we can talk about frogs, specifically wood frogs. This is the species that I am interested in for my PhD. So just briefly, wood frogs are an amphibian. And on the bottom there, you can see a photo of the adult form of this species. So these frogs breed early in the spring. And if you live around here in Connecticut or in the Northeast, you might hear them in the spring. They have this call when they move to the ponds to breed. They really sound like ducks quacking. And I'm also showing you here a photo of what a clump of wood frog eggs look like. So these are laid in the ponds. Eventually, these eggs will develop into little tadpoles, as you can see in the photo I have here. These were tadpoles that I caught in Alaska, actually. And about 2 months after these tadpoles emerge out of their eggs, they will metamorph. So they will change into their adult form and leave the pond and move on to the terrestrial period of their lives. Next slide, please. All right, what makes this species incredible is that they have this really interesting freeze tolerance ability that they undergo during winter. What makes them different from other animals, say like bears that hibernate through winter, is that these frogs completely shut down. So they don't have any heart rate, no brain activity, no muscle movement. And if you see them, they will probably look like they are just suspended in time or even dead, but they are not. And if you try to hold them, they will feel like a solid block of ice. We like to call them frogsicles. And these two frogs are specimens that I have collected from a forest here in Connecticut that I brought into the lab, and I put them through a freezing trial. And after a couple of days, I slowly warmed them and they actually completely recovered. They were completely back to normal. And I find that just absolutely incredible. Next slide, please. So there's a very interesting biological process to how these animals have managed to do this freeze tolerance and how they have this ability. And I'd like to mention that they are one of very few amphibian species and very few animals overall that have this ability. So if we zoom in into the body of the wood frog, I have here just a very simple schematic of a few cells and then the outside environment of the cell. Sometimes you would hear it referred to as the extracellular space. And in the cells, for any animal, whether it be humans or the wood frog or even insects, if your cells freeze, if ice starts developing inside the cell, those ice crystals are very detrimental. They will injure the cells and that will always cause death to the organism. How the wood frogs manage to bypass that is they produce a lot of compounds such as urea, glucose, which is a sugar molecule, glycerol as well. And as they increase more and more of this compound, they increase the concentration of it inside their cells. That makes the cells unfreezable. However, the space outside of those cells can and does produce ice. That's where that solid ice formation happens, and that is completely normal for the wood frog's body. And about 60 to 70% of the water content of the wood frog's body turns into ice. Next slide, please. All right, the other really interesting thing about wood frogs is that they have a very large range. So a range is just what determines where we naturally find these the species in the wild. So I have here a map of the United States and Canada, and in gray is the region, the current known region, range, of the wood frog. So this is where we expect to find them. As you can see, they cover a very large range. In the south, their limits are around Georgia and Alabama, and they go all the way into the Arctic parts of Canada and also in Alaska. I've highlighted a few regions where we have evidence and we have data of what the tolerance limits are for these frogs. So these are all studies done by other researchers. And what I'd like to highlight is that there's a lot of variation in the tolerance limits. So for example, in Ohio, the wood frogs can survive minimums of about -2 to -3 degrees Celsius of freezing. But if you go further north into Fairbanks, Alaska, we have evidence that these populations of wood frogs can withstand freezing at -18.1 degrees Celsius, and they can stay frozen for about 6 months. And there's an area that I highlighted as unknown. This is of particular interest for several reasons. One of them is that these are the populations that are north of the Arctic Circle. So wood frogs are the only frog found north of the Arctic Circle in North America. We know very little about those populations, and I will highlight later why they're also interesting for a different reason. Next slide, please. So once again, another map of the United States and Canada, though parts of Alaska are missing. And this overlaid here are climate zones as determined by the USDA. And all I want you to know here is that all these different colors correspond to a different zone and that's determined by the minimum temperatures. And you can see as we go further north in latitude, the climate gets colder and colder. And these arrows are showing what we think the historic movement of wood frog populations into the continent. So just to take us back a little bit, thousands of years ago, 15,000 years ago, this part of the world was covered with ice. That was the last ice age. And after all that ice receded, there were wood frog populations in these areas that I've marked with letters. And this information about this movement was conducted by a researcher in 2008, Julie Liao and colleagues. And basically, when all that ice receded, these wood frog populations that were further in the south were able to start moving into colder and colder areas. They were moving more north. You can see that the arrows generally point in a northward direction. And I'm very interested in that historic movement and how they were able to move into new environments, colder environments generally, and how they have adapted to those conditions. And with my PhD work, I'm hoping that if we can understand some of that historic movement and the abilities of wood frogs to adapt, that we might be able to understand their abilities to adapt to our currently changing climate. I am also very interested in the edges of these populations. For example, the Arctic is warming at rates much higher than the rest of the world. And so you can imagine there will be challenges for these wood frogs that are tuned to stay frozen for longer periods of time. If there's any disruptions to their winter, to the winter time in these regions, the wood frog might not freeze for as long or might freeze and thaw multiple times. And all of these processes are energetically costly to the frogs. So one way I am trying to answer these questions is through looking at the genetic variation of wood frogs populations. And I am trying to look at that across the range, which is a very large range. And to obtain data about the genetic variation, we need to collect actual tissue samples from these animals. So that can be things like liver or muscle tissue, and process that tissue to obtain genetic information. Next slide, please. So how do you collect tissue samples? Well, we have to go and look for animals. So this is a photo of me and three of my friends, Kaylin, Andis, and Sam, last summer in 2021. We were able to conduct this fieldwork trip to the Northwest Arctic Borough in Alaska. This is the region that I've highlighted before, that is in the Arctic. We were especially interested in capturing the genetic variation of this region because it is one of the areas at the kind of edge of that range. But also, if you remember that historic movement I have talked about, the populations here are expected to be the furthest away from those southern populations at the time of the Ice Age. And we expect to see some interesting patterns of genetic differentiation that I really wanted to understand. Next slide, please. So the left is a photo of me and my friend Andis. We were looking for frogs and tadpoles in this beautiful Alaskan wetland. And on the right is what doing fieldwork in a remote area looks like. So we packed all of our essential supplies to collect and process tissues. You know, there are no benches, no lab space. So we were on the ground and working out of tents. So you might be thinking, this looks pretty fun. It looks really cool. You get to go camping and look for frogs all the time. So just go ahead and do that for the rest of the range. And what I would love to do that full time, it's absolutely impossible for me to collect tissue samples from that huge range by myself in 5 years, which is the time that I have to do my degree. Next slide, please. And this is where the museum collections have been essential, have made this project that I'm trying to do possible. Once again, this is a map of the United States and Canada, and in the purple overlay is the range of the wood frogs. And all these circles and squares represent tissue samples that I have of wood frogs. And about 80% of them are samples that I've been able to borrow from museums, primarily the Peabody Museum, which had a really awesome collection of wood frog tissues. But I was also able to reach out to institutions in the United States and also in Canada and ask for wood frog tissue samples to use. So I have here a photo of 2 wood frog specimens that my colleagues and I collected on our trip to Alaska. And we preserved them in a way that will make them well preserved for years and years to come and put them at the museum. And just personally for me, it's very important to know that I'm contributing to this collection that I am heavily using and doing my part for future researchers to also use this data that I'm collecting. On the left are pictures of tubes, and this is what I primarily work with. So the museums have collections that could be whole specimens that are preserved, but what I usually use are little tissue tubes, and they contain tiny pieces of wood frog tissue, which could be liver, muscle, toe clips, or tail clips of tadpoles. So once we transition into the demo version, I will talk to you a little bit about what happens next. What do I do with these tubes to actually translate them into usable data? And that is it for the slide portion. I think we might be able to take some questions before we transition. Thank you all for listening.
Yara, we have a question. Are wood frogs able to adapt to higher temperatures as well as lower temperatures?
Yeah, that is a very interesting question. And we've actually done a little experiment. I did a collaborative experiment with a colleague, Andis, where we were looking at thermal performance. So we were quantifying the jumping performance of frogs as we were exposing them to higher and higher temperatures. So they absolutely could. They are called adapted species, but they might be able to. And this is where looking at the impact of climate change is really important because especially for more of the southern populations in Alabama and Georgia, they are already at regions where it is much more mild than the other populations further north. So looking at what are those populations already adapted to warmer temperatures, because it doesn't really get to freezing or sub-freezing temperatures down there.
Okay, another question. Can wood frogs withstand being repeatedly frozen, then defrosted, then refrozen over the course of a winter?
Yes, great question. They do. So one thing that previous researchers were looking at is that pattern of freeze-thaw, because the wood frogs do undergo multiple freeze-thaw cycles as the temperatures drop and cool and warm, they do go into multiple cycles, and every cycle is energetically costly. They expend a lot of their energy and their glucose reserves to go through these cycles. So one question that researchers have been debating is whether these multiple bouts of freezing and thawing might be useful or negative to the wood frog. And we still don't have a clear answer about that.
And a sort of related question, it says, must wood frogs freeze to complete their life cycle?
I have that same question. My working hypothesis is that some of the more southern populations don't go through any freezing. We don't have any concrete evidence. No one has actually performed a freezing trial on frogs that were naturally found in the southern regions of their range to actually determine whether that is possible or not. But I have looked at yearly temperatures at those regions, and it doesn't really get that cold, especially because the frogs, they burrow under the leaf litter about 10 centimeters below the soil. And I do not think that it reaches those cold enough temperatures for them to freeze, or at least freeze for a long time. But I cannot determine whether that is true or not yet.
Okay, we have a few more questions coming in, but I think we'll move forward with it with the lab demo, and we'll get to these questions in a little bit.
Sounds great.
While Yara is getting set up for her demo in the lab, I'd like to take this opportunity to share some information about Frogwatch, which is a program that the Peabody Museum has been a part of for a number of years. Frogwatch is an all-ages citizen science program that trains volunteers to identify frogs by their call. Once they're trained, individuals can choose a local wetland and then monitor and report on the species that they're able to identify. The Peabody collaborates with the Beardsley Zoo and the Norwalk Maritime Aquarium to do the training, which is now done all online. And training dates are coming up soon on February 24th, March 9th, and March 15th. If you'd like to learn more about the program or to register, you can visit www.maritimeaquarium.org/citizen-science. And that link will be available in the chat for you if you'd like to pursue that. Now we'll be going back to Yara in her lab.
Hello, welcome back from the bench. So this is where you'll find me most days. These days, I have been going through many samples to process them, to get them to that usable data that I talked about. So how I usually start is with just a small tube that contains a tiny piece of tissue. This could be as small as a lentil piece or even smaller sometimes. And that's really all we need to generate incredible data. And The basic process of doing a DNA extraction is actually pretty straightforward. It's like following a recipe. What we start with is the tissue sample. We use these specific solutions that will break up the tissue into smaller pieces. It will lyse the cells. It will break them open. It will break down proteins and enzymes because we want to release that DNA into our tube. So the next step usually looks like something like this. Sometimes if I'm starting with a muscle tissue, what we call the lysate will look like this. Sometimes it looks a bit darker because this was a liver tissue. So now everything is broken up into the solution. Our DNA is here, but so is everything else that was in the tissue, and we don't want that. We want to just have purified DNA, so we don't want anything else. And that's where we use this other tube. And if you can see here, there is a little white filter. So the technology of this tube is that filter will bind the DNA. So I would put my lysate into here, and the DNA will stick to that filter. It has a special composition that would hold on to that DNA. So once I do that, I will use various solutions to wash away everything else. So you have your DNA on that filter, and then you will add that solution. Put this into a centrifuge, which is a device that will spin it really, really fast so that the liquid will get pulled to the bottom. So we do that a couple times so that everything that is not DNA will come out. We get rid of that. Finally, we use another special reagent that now will bind the DNA. So now that we get rid of everything on that filter, now we want to take the DNA from that filter into the bottom of the tube. This is what the final product looks like. It is not very exciting because it just looks like a clear solution. But if you believe me, there is DNA in here. Ideally, this will be as pure and as clean as possible. So no other contaminants, no enzymes, nothing like that. So once I do that, the last step of this process is putting the DNA into an even smaller tube. And I use this lab device that uses fluorometry. So it will be able to tell me how much DNA is in this sample. So I'm just going to show you a sample that I ran a few days ago. So it's very easy to use. It will tell me just the concentration of the DNA. So that's kind of my guarantee that the process went okay, that I was able to extract the DNA. After that, I repeat this many, many times. And after I am done, this is where I will send these samples to a sequencing facility. And what happens at that facility is they will take all my samples, put them through machines that will translate that genetic information into a giant text file. And then they will send me all those files, and I will sit at my computer and start analyzing that. And what the information in those text files will tell me how differentiated every sample is from the other. And using that, I'll be able to ask questions about how differentiated are these populations that I'm interested in. Can I see any signals that might point to them being able to adapt to different environmental variables, such as temperature and rainfall pattern and snowpack? So this is essentially it. This is what I do for my lab work. And I think we can answer some more questions about the lab work or the slides.
Okay, Yara, we have a lot of questions for you. The first one is, it says on the map of tissue collections, your last slide, what do the different symbols represent?
Sure, yeah. The black and yellow were samples that we've acquired through the museums. And the red ones were samples that I either personally collected or I have collaborated with other people so that they can personally go out and collect for me in harder to reach regions. So a lot of folks helped collect last summer in some regions in Canada, British Columbia, in Norman Wells, which is was a really exciting location for me to get data from, and some other regions in the United States too.
Okay, every year for 30 years, I have wood frogs mating and laying eggs in my garden pond. This year, the eggs disappeared in a few days and only a few tadpoles, and they disappeared. What can I do to protect them?
Oh, that is very exciting. Yeah, that's a great question. My first, the first thing that comes to my mind is don't do anything to interfere with the pond wood frog, the wood frog ponds. So my lab has been monitoring several, not even several, but 40 plus ponds across Connecticut for over 20 years. And we've seen that every year is different. Sometimes we have what we call these extinction events. Sometimes they're because there is a ranavirus infection in the ponds. So this is a very deadly virus for the frogs. And sometimes they just die off and we don't know why. But they have these different waves and they bounce back sometimes. So I think they're very resilient. Yeah, so I wouldn't do anything to protect them. But I would advise you to try and join the Frog Watch because that data is really cool. And I know my lab would be very interested to know and learn about those patterns of different ponds that we don't really usually monitor. It's very interesting.
And another question. How does the rate of freezing affect their ability to survive being frozen?
The rate of freezing, interesting question because I have had some thoughts about that too. Because all of the data that we have, most of the data that we have from researchers who have been doing this since 1980s has been controlled experiments in the lab. And we know that when we freeze them in the lab, we have to do it very slowly. So what I do is about 1 degree Celsius a day. And we don't really know how rapidly it happens in the wild, right? It's very hard to monitor temperatures where frogs are in their hibernacula, so the sites that they choose to go in for freezing. So we don't really know how rapidly it happens, but my guess is that whatever they can tolerate in nature is probably they can tolerate, but we don't know how rapid is too rapid for them.
Okay. A question about the frog's predators. It says, what are these frogs' predators, and is being prematurely thawed make the frogs more vulnerable to them?
Yeah, so a lot of things eat these frogs, especially the tadpoles. So they actually have a very high mortality rate when they're tadpoles. So that just means that most of them die before they can actually escape the pond and metamorph into adults. So when they are, even before they're tadpoles, when they're still eggs, a lot of salamanders like to munch on them. When they're tadpoles, they're attacked by a lot of invertebrates in the ponds. So things like dragonfly larvae, they're very great at hunting tadpoles. And when they are in their adult form, a lot of snakes eat them. And in the winter, premature thawing, we don't know a lot about what happens in the winter because it's very difficult to find frozen wood frogs that are under the soil. I would guess that they will be pretty vulnerable if they are thawing prematurely. But we don't really know.
As kind of a technical question here. What parts of the genome are you targeting?
Yes, love that question. I am doing dd-RAD sequencing. So this is a reduced representation of the genome when we're doing genome sequencing. So the DNA sequencing, you can do a full genome sequencing. So this is getting all the information of the genome. It is very expensive to do that for 100+ samples. So I'm doing this reduced representation, as the name implies. It gives us a reduced view of the genome of these animals. And how this works is that it will give us the same region for all the different samples, so that we can actually compare them, right? If you have different regions that are not matched between the different samples, that would be hard to compare.
A question about collections. It says a lot of outside critics of collections state that they encourage researchers to take more specimens than needed because of the mindset that we must get more for the future. How would you respond to this?
Yeah, I think that's a very valid question. So one thing that I have learned through doing this research is the process of going through permits and how we can actually have permission to do the work. And there are multiple channels that will make sure that we're really only doing the work that is necessary we're only collecting what's necessary to do the work. So for example, we've asked for permits to work in Alaska and specifically in the Kobuk Valley National Park. And we had a lot of back and forth with different people to ensure that we're really collecting what we need and we're not just collecting more for the sake of collecting. So I've had that experience with permits there, but also like all my other permits in different states and in Canada as well. So I was actually very not surprised, but it was good to know that people do care and they want you to do the work, but do it right and really not cause harm.
Sort of a related question says, when you collect samples, are there efforts made to keep frogs from feeling pain during the process?
Yeah, absolutely. So the other skill that I have developed when I started working with animals is working with the institutional animal care. The acronym is ICUG. And they are wonderful to work with. Again, they also want to ensure that we are doing the science that is necessary, but really trying to minimize harm as much as possible. So, talking about the specific protocol, the animal treatment, where you keep them, and also the numbers of animals. So you have to really be thoughtful about how many you really need to do the work. So I think it's a great skill that I was able to develop and learn about as I was doing all this work.
And one last question we have here. It says, on your map of the end ice age populations, location A is a surprise considering that the area was covered by the continental glacier. Please explain.
Yeah. So yeah, I'm trying to remember where that A was. So that was where we think the refugia populations were. So after that ice receded, the remaining wood frog populations seeked refuge there, and that's where the current known populations of wood frogs originated from. That as
That's it. That sounds good to me. One other item, it says that, and I did not know this, you work at the museum, and someone's asking what exactly your job is.
Yeah, so I have a pretty cool part-time job as a museum assistant. I specifically work at the vertebrate zoology department with Britt Watkins Caldwell, and I do a lot of processing samples. So we get a lot of samples sent here, and there's a lot of work to make sure that the data files that were sent match what we actually received. putting numbers into sheets. We label every tube with specific number, so putting different tags on them, and we put them in our cryo collection, which is our super cold freezers that will really preserve the tissues for years to come. So I do a lot of that.
Okay, I think we've gone through all the questions. I want to thank you personally, for an incredible presentation, and thank you to everyone else who's joined us this morning. We hope everyone will follow the Peabody on social media and visit the Peabody's MLK event website to learn about our other upcoming programs throughout the holiday weekend. Take care and stay warm. Bye-bye.